Great. Well, thank you very much for the opportunity to be here. Really excited to share with you what we're doing at Senti. We're basically a synthetic biology company that's focused on creating smarter cell and gene therapy products, ones that we can control the precision, the safety and activity of in vivo. So here at Senti, we are laser focused on taking our lead programs into oncology and showing that this gene circuit technology that we've been building for the last few years has the ability to really transform the way in which we treat cancer. The central challenge that we're aiming to overcome in oncology is that many existing therapies that we have today are either not broad enough to completely overcome the heterogeneity of cancer cells, as well as not specific enough to uniquely just target cancer while limiting off-target toxicity. And as a result, that narrow therapeutic window ultimately leads to a difficult treatment paradigm for many oncology indications today. So to overcome this issue, we've developed a technology called logic gates, which I'll walk you through in a little bit. But this allows us to essentially target a heterogeneous population of cancer cells while at the same time protecting healthy cells from being killed. And if successful in the clinic, we think this has the opportunity to really be expanded beyond our initial indications, which are really focused on AML, and able to address a wider range of oncology indications, including solid tumors. Senti 202, we are moving towards the clinic. The IND is on track for this year, and we aim to get this into clinical studies as soon as possible. So just as a highlight, Senti 202 is a program directed towards the treatment of acute myeloid leukemia. This is a disease that has a high unmet need. Relapse refractory patients unfortunately have a median survival of about 5 months. AML is a disease that's pretty heterogeneous, where there's no single target out there like CD19 or BCMA that you can use to wipe out the entire population. So what we've done is systematically engineer Senti 202 to actually go after multiple targets found in AML, and at the same time protect any healthy cells that are expressing those targets, including the bone marrow stem cells, from being killed. And this is to really address one of the key challenges with prior AML therapies, which is that many of the targets that people want to go after in this disease are also found on those healthy stem cells. We are using a CAR-NK backbone for this product. We think that makes a lot of sense given that NKs on their own have activity in AML, completely unengineered, and we're building upon that sort of native killing activity of NK cells to achieve greater cytotoxicity and to simultaneously go against AML blasts and leukemic stem cells. Before I get into the program, this really came out of this platform that we've built at Senti to allow us to build what we call gene circuits. Gene circuits are essentially multi-gene components that we insert into cells, allowing us to program cell and gene therapies to do things like logic, which basically refers to sensing and responding to multiple antigens. Multi-arming, so we can engineer a single product to do 2 or 3 or 4 different things at the same time. What we call the regulator dial, which is essentially a small molecule switch that we can use to turn on and turn off what the product is doing using FDA-approved drugs, as well as smart sensors that allow us to program when, where, and under what conditions a particular cell or gene therapy should be active. We have used this platform to enable a variety of portfolio programs in oncology, Senti 202 being that lead program. That's really what I'm going to focus on today, given the limited time. That being said, the gene circuits do work in both NK as well as in T cells and for a variety of other applications. Senti 401, for example, uses our logic technology in solid tumors, going after CEA as one of the key targets and protecting healthy cells from being killed. Senti 301A is a program directed towards GPC3, potentially for treatment of liver cancer as well as other solid tumors. We've also been very excited and fortunate to have collaborations with some of the leading companies in areas outside of oncology. So with Spark, we've been developing very specific promoters, compact in size, that allow us to target very specific on-target cells for activity while minimizing any off-target expression. And with Blue Rock working very closely to insert our gene circuits into iPSC-derived cells for regenerative medicine applications. So ultimately, these gene circuit technologies are portable across modality, very similar to genome editing in that sense, and we have the ability to go beyond our own internal modalities to support other partners. So let me tell you a bit more about what Senti 202 does and why we're so excited about it. So Senti 202 is a CAR-NK program where we're leveraging peripheral blood-derived NK cells from healthy adult donors. There's 3 components that we're engineering into the product. So the first one is in the upper left-hand corner here. It's an activating CAR that's designed to go after 2 targets simultaneously. The way we do this is through a bivalent CAR design. It basically targets CD33 as well as FLT3. And I'll go into some of the biological rationale behind this a little bit later. But both of these targets are well validated in AML. CD33 tends to be expressed on the blast population, while FLT3 is found on the leukemic stem cell population. So by going after these 2 distinct populations in patients, we hope to drive deeper and more durable responses. Now, that seems sort of like an obvious idea. Why not do this with other products like an ADC or a single target CAR? It turns out that both CD33 and FLT3 are found on healthy tissues. So in particular, if you treat with CD33 or FLT3, you oftentimes will suppress the bone marrow as well as many of the healthy blood cells that we're trying to, the bone marrow naturally produces. So ultimately, that limits the therapeutic window for these sort of approaches that target these AML targets. To solve that, what we did was design what's in the middle here, this purple receptor. This is what we call an inhibitory CAR. What it does, instead of activating the NK cell, it basically tells the NK cell to shut down. It's basically a do not eat me sort of signal. We designed this by basically designing a large library of inhibitory CAR architectures and then screening and optimizing through repeated design cycles to find an inhibitory CAR that can give us good protection of healthy stem cells. It does this by recognizing a particular target called EMCN or endomucin, which is highly expressed on those healthy cells, not expressed on the cancer cells. So it's basically an inverse relationship with the tumor targets that we're going after. Finally, we've engineered a calibrate release IL-15. This is our own proprietary version of IL-15 that gives you both membrane-bound activity as well as secreted activity. So it gives you sort of the best of both worlds in terms of IL-15 signaling. Our plan for Senti 202 is to go into relapse refractory patients with in AML as well as MDS that express CD33 as well as FLT3. Given this good safety profile associated with NK cells, we are planning a multi-dose, multi-cycle strategy here where lymphodepletion followed by multiple rounds of Senti 202 dosing can occur. One of the things about AML that's attractive from a clinical development perspective is if the product's going to work, we believe that even within a 28-day first cycle, you can start assaying the patients for responses. And these patients can qualify for additional cycles downstream as well. This being a phase 1 study, we're certainly going to look at safety profile, but we are actually also going to simultaneously look at biomarker responses. Are we killing off CD33, FLT3 positive cancer cells? Are we sparing those healthy tissues? As well as signs of efficacy, including response rates in these patients. So just to highlight how we designed this product, Senti 202 from the beginning was designed to address known biological challenges in the treatment of AML. AML has been a really challenging place for drug development in general because of the disease heterogeneity that's inherent to this set of patients. There have been single-target ADCs as well as CARs developed in this field. Against CD33, for example, Mylotarg is an approved CD33 ADC product. There have been also CD33 allogeneic CAR NK cells where people have seen good upfront complete response rates, but relapses, unfortunately, in these patient populations. Similarly, there have been other CAR NK therapies going after, for example, NK2GD ligands that have had some similar issues. And as you look into the biology, I think a lot of this we believe can be attributed to the fact that these single target approaches are essentially missing a key portion of the tumor population, which are these leukemic stem cells. Leukemic stem cells are a pretty rare population of AML cells, but have now been shown in a variety of publications, both in preclinical as well as looking at clinical data, to be really a nidus of relapse of cells that basically escape treatment and can basically regrow and repopulate. So to address this issue, like I said, we've engineered Senti 202 to go after to contain 3 killing mechanisms. One is leveraging sort of really good, well-activated NK cells to do a baseline level of killing. CD33 tends to be expressed quite well on the AML blast population, and FLT3 allows us to address that leukemic stem cell population. So putting all 3 of these together, we believe we can increase the overall activity of the product. As I mentioned earlier, the issue with these targets, though, is that they're also expressed on healthy hematopoietic stem cells. And so that leads to a limited therapeutic window due to this off-target targeting approach. This is where our NOT-gate technology, using that inhibitory CAR, allows us to protect those healthy cells from being killed. Oops. So we have shown across a variety of AML patient samples, these are all patient-derived blast and leukemic stem cell populations, that when we engineered Senti 202 circuit into NK cells, we achieve really significant and much more robust functionality and anti-tumor killing across these entire populations that are pretty heterogeneous and come from different AML subtypes. We've also shown that this does translate into more durable responses in a variety of in vivo models. We have long-term survivors in these models past 200 plus days, showing that the overall sort of depth and activity of these products can be much improved. And now we have also a variety of evidence showing that Senti 202 does allow us to indeed preserve the function of hematopoietic stem cells. I think the data here that I like to call out just in this period of time is the one in the middle here. This is looking at colony-forming units that are generated by hematopoietic stem cells. This represents their ability to regenerate and populate all the downstream blood lineages. And even with treatment with Senti 202, we still see a quite robust maintenance of this colony-forming unit process. The other data also shown here shows that Senti 202 does have this ability to enrich for these healthy cells in vivo at the expense of killing cancer cells. Finally, we have shown that our calibrated-release IL-15 does enhance the overall persistence of our product, both in vitro as well as in vivo. It leads to a greater enhanced overall length of time that Senti 202 can persist, as well as a higher increase in the amount of cells that can be found in the blood. And certainly, we can redose patients with this product as well, given our manufacturing approach. So we're very excited again about Senti 202 as it moves through the IND process and into the clinic. We're leveraging the fact that these NK cells do have this native anti-tumor killing activity, the fact that CD33 and FLT3 are well-known targets, so we can address these directly in a single product, and also leveraging that inhibitory CAR to protect healthy hematopoietic stem cells from being killed. There are additional programs in our pipeline that essentially leverage some of these components that I mentioned earlier. So Senti 301A at a high level is targeting liver cancer using GPC3 as a target. Actually, GPC3 is a pretty clean target by itself. NK cells also get to the liver quite effectively. So we think combining these two together, in addition to arming this with our calibrated-release IL-15, has the potential to drive deeper and stronger responses. We are pursuing potential strategic partnerships here to do first-in-human studies, potentially in Asia or other places where liver cancer is highly prevalent. We have shown a variety of models that actually our Senti 301A construct is quite active. There are other trials that have used GPC3 as a CAR T target where they actually have seen good clinical activity, but some challenges in terms of the safety profile. And we believe that the CAR NK platform has the ability to overcome some of those key challenges, given that the NK products generally have been much safer than CAR T programs. Finally, I mentioned Senti 401A. This is leveraging actually that same logic gating technology, but addressing colorectal cancer. We are going after CEACAM5, which is a known target again in solid tumors. There's a phase 3 CEA ADC being in development now against this particular target. The issue with CEACAM5 as a particular target is that it's also expressed on healthy epithelial cells, and this has manifested itself in a variety of toxicities that have been seen in clinical studies. So what we did here was take our inhibitory CAR architecture and really adapt it to go after VSIG2, which is an antigen that's highly expressed on those healthy cells, but not expressed on those cancer cells. So again, using this same inhibitory CAR architecture to protect those healthy cells from being killed. This product contains also IL-15 plus IL-21, which together we've shown actually overall boost the activity of the product against solid tumors. This is shown here on the left. It leads to essentially a more durable response. And if we actually look at the ability for IL-15 and IL-21 together, it actually allows these cells to resist immunosuppression by factors such as TGF-beta. In the remaining time, I think this is actually kind of a fun set of movies to look at. This is really looking at that logic gate activity in action. So these are 3 imaging experiments that we've done where we mix cancer cells and healthy cells in close proximity with each other. Cancer cells are shown here in red, while our healthy cells are shown here in green. So without any NK cells, what you see is that both the cancer cells and healthy cells grow, as you might expect in this sort of experiment. Now in the middle here, these are CAR NK cells that actually only have the CEA activating CAR. And what you're going to see here is that both the cancer cells and the healthy cells get killed because essentially both of them express CEA. Finally, on the right-hand side, these are our Senti cells that have our NOT gate technology. What they do is they recognize VSIG2, which is present on these healthy cells. And you can see pretty clearly what happens is that you can suppress the cancer cells and kill them, while you can preserve the healthy cells and allow them to survive. I think this is a really good demonstration of the engineering that's gone into this, because all these cells are actually in really close proximity to each other, and so this shows that our CAR NK cells are really able to differentiate between even neighboring cells that have healthy or cancer phenotypes. This platform has been translated also into T cells, and we think there's a lot of opportunity for this to actually play a role in CAR T therapies that are being used for a variety of applications as well. So that's actually what the last slide showed. So just to finish up here, I'm running out of time, but we have shown that our gene circuit technology platform actually works across a variety of different cell types. We're advancing our Senti 202 CAR-NK program through IND and into the clinic soon thereafterwards. And we have validated actually this technology, as I said, across multiple modalities. Quite interested in exploring partnerships with other companies. If you have a cell type or a particular application of interest where essentially more sophisticated genetic control would be helpful to increase the precision or the effectiveness of those products, Senti can basically deploy our gene circuit technology in those areas. This includes things like T cells, AAVs, and into iPSCs as well. So with that, thank you very much for your time, and looking forward to connecting with folks. Appreciate it.